Loud?
Okay, hello everybody. We are now doing session 5 of neuroanatomy. And I was just saying while we were all gathering that we're going to shift gears today and for a good part of the course now. The first 4 lectures were introductory, and they were at a nice and easy and relaxed pace, and we didn't have a lot of details. We were looking at whole concepts and things like that. But now, and I mean now, we're going to shift gears to a fast-moving and very full set of lectures. So if I can make this work. Right, okay, here's an outline. And just from the length of the outline, you can see how many topics we're going to be dealing with, and all of them in detail. Just to set this, though, where we're going to do is we're going to start at the spinomedullary junction, and then we're going to go up to the pontomedullary junction. So the entire lecture is on the medulla. We're going to look at many different levels of the medulla axially, but we will always be in the medulla. And this is entirely devoted to the medulla, but including its connections. We'll have to talk about other parts of the brain. For those of you who are trying to learn from this series, this is a strong reminder that you need to spend at least an hour a week studying these slides and using what other reference resources you have to understand this material. There's no way to grasp all of this in an hour of this talk. All right. At the spinal, let me also say that we don't have a lecture. The question would be, why aren't you starting with the spine? Like, where's the spine lecture? The spine lecture is going to occur when we talk about motor systems and sensory systems, and there is no lecture on the spine, okay? At the spinomedullary transition. So first of all, and we're just talking, what do we see? You're gonna see the corticospinal decussation. You're going to see the posterior columns and their nuclei, which are going to then be associated with the decussation of the medial lemniscus. We're also going to be looking at this accessory cuneate nucleus, which is an important player in our proprioception and motor coordination, and sits above the dorsal nucleus of Clark with an analogous purpose. Both of them get proprioceptive input from dorsal root ganglion cells, and then give rise to mostly uncrossed fibers to the ipsilateral cerebellum. We're going to look at the exceptions to that as well. The Clark's nucleus is part of the spinal cerebellar pathway, and it's for the leg, and the accessory cuneate provides the same purpose. It creates the cuneo-cerebellar pathway from the arm. Both of those are going to enter the cerebellum, usually ipsilaterally, via the inferior peduncle, which we will look at in quite a bit of detail. And then there's this very peculiar little nucleus called the arcuate nucleus, which is going to explode, but simply explode as we enter the pons at the end of the hour. So here we are. Could someone turn the lights off, please? All right, so here we are, and let's just start at the top. All right, and a lot of looking from now on, looking at where these things are. So here's the posterior columns, right? The fasciculus gracilis and cuneatus. This is a beautiful slide. Of course, this is a Weigert stain for myelin. And it shows you the separation between the fibers for the legs and the fibers for the arms. So they're really quite different, separate from one another. We are at the spinomedullary junction. And so here's the dorsal root of cranial nerve, of cervical 1. All right. And here's going to be the ventral root down here of the ventral root of cervical 1. Now, the substantia gelatinosa is a upward extension from the spinal cord, and we will be dealing with that when we talk about sensory systems. Here's cranial nerve 11, way in here. Okay. This is the area, the central gray and the central canal. Here is the anterior. I'm going to use the term ventral spinal cerebellar tract. Doesn't matter. You could say anterior posterior or ventral dorsal to be consistent with the rest of this series of talks. I'm going to use the term ventral and dorsal. All right. And then buried in here is the spinothalamic tract. Right. Here is the sum of the corticospinal decussation right here in the raphe, in the midline raphe. All right. Here is the MLF. And I expect all of you to go, what? Okay, isn't the MLF midline up high? Doesn't it connect the 8th and 6th complex to the 3rd nerve complex? What is it doing down there? And it's like, hello, the MLF. So my first question is this ascending or descending? What's going on with this piece of the MLF? I looked to Shruti, who, if anyone's going to know, it's going to be Shruti. Yeah. This is a descending portion of the MLF. And its purpose is going to be to connect vestibular information and reticular information, and we're going to talk about reticular a lot, with the neck muscles. Because when you turn your head to look at something, you better be coordinating your neck muscles and your eye muscles, or you're going to be way off. And that's what this is about. So this is a descending MLF pathway, and it's considerably larger than the ascending MLF. Here's a vestibular spinal tract, which is right next to the MLF, which now that makes sense because this is vestibular into the neck for vision. This is just plain old vestibular into the rest of the spine. Here's, as we said, ventral root for cervical 1. Here is the lateral corticospinal tract, which has moved away from its very peripheral site in the cord. And it's sort of nice. Look at it here. And it almost looks like a pontine nucleus running through the basis pontus. And it should, because it's the same structure. But of course, it's moving medially now because it's in the about to decussate, or just did decussate, and is now going to be heading out to that lateral. Here is the the dorsal spinocerebellar pathway. And here's a little dorsolateral fasciculus, which is an extension of Lissauer's tract, which we will talk about again when we talk about sensory systems, and is closely related to the substantia gelatinosa. So we are almost sort of cervical in this slice and almost sort of medullary. And by the way, down here are the your axial locations that we'll be using, and I give you this for every section. All of these sections and these Weigert stains are from Truex and Carpenter 8th edition, and are, in my opinion, the best available atlas that you have of brainstem. All right, let's moving up a little bit now. Okay, moving up, and we start to introduce the nucleus gracilis. All right, and then we're going to talk about the spinal trigeminal tract and spinal trigeminal nucleus. We're going to talk about those in a minute. So I'll come back. I want you to see where they are. Still got central gray, still got a ventral spinocerebellar, still got spinal thalamic. The corticospinal decussations are in full bloom up here. There you are. Right, some of it right here. And as a result of that, you're now seeing pyramids form on the anterior surface of the medulla. You have, again, the vestibular spinal, the medial MLF. Here is a supraspinal nucleus. What is a supraspinal nucleus? Actually, it's the anterior horn cells of cervical root 1. Okay. Here's cranial nerve 11. We're going to look at this in lots of detail. The dorsal spinocerebellar. Here's nucleus cuneatus. Right? These are going to become very big on the next slide up. So what will be happening, of course, is that the fasciculi will be getting smaller and the nuclei will be getting bigger as the posterior columns terminate in their second-order neuron pathways. This slide is merely to show you the accessory cuneate nucleus, which we will talk about, but here's its location, and here's Clark's column. And this is a good slide to show you. So this would be the area of the, so dorsal root, ventral root sort of thing. These will be your anterior horn cells, your supraspinal nucleus. This is the dorsolateral fasciculus will be up here, and the substantia gelatinosa will be here. But in future talk, we will look at that in great detail. All right, let's talk about the spinal trigeminal complex. And let's get rid of the idea that this is the spinal trigeminal tract or anything like that. It's not. This is not an upward extension from the spine. This is actually a downward extension from the trigeminal nerve. and its nuclei. All right. The afferents that are going to the spinal trigeminal complex, it's a trigeminal complex is what it is, and it happens to be in the spine. Okay, that's the way to think about that. So the afferents are coming in from the trigeminal nerve, which is all over the place, and going to be mostly phase, of course, pain and temperature from phase. They'll be mixed in with some pain and temperature fibers from 7 and 9 as well. But this whole complex is enormous, as you're going to see, and extends from really from the midbrain all the way down to seat 2. This has clinical importance because at the cervical medullary junction, sometimes you will have a suspended sensory disturbance of the forehead and upper portions of the face, and it's not clear to you why. And if you're not aware of this complex, you're going to look for a second localization, which isn't there, right? And the other part of this is it's somatotopic, but it's upside down. I borrowed this emoji to make this point. And that is, the more caudal you are in the spinal cord, the more frontal it is, okay? And as you move up toward the medulla, you get toward the chin and mouth. It corresponds to the laminae of the dorsal horn. It's organized as the dorsal horn is in an onion skin kind of way, layer upon layer upon layer. We will study that in our talk on sensory, incoming sensory. But right now, I want you to just again take away that this complex carries pain and temperature from the ipsilateral face, but then projects to the contralateral thalamus. And therefore, the spinal trigeminal tract would be better off being called the trigeminal thalamic tract, because that tells you what's going on and avoids this confusion of it being a spinal anything. It's not. It's simply, it's so big that it goes down into the spine. And that's all there is to it. It's not connected to the spine in any important way. All right. This is a good slide. So here you are, and here you are in the Gisserian ganglion, semilunar ganglion, trigeminal ganglion, whatever you want to call it, with its 3 divisions. And here you can see the dorsal root ganglion, and you can see all of this information now is going to be coming from all over the face. All the divisions are going to be funneled in through here, and then they're going to splay out. Some of them are going to go, we will still be studying trigeminal nerve up in the midbrain, okay? We have, just for orientation, here's the motor nucleus of the trigeminal nerve. Not very big, okay, but important, of course, mastication, that sort of thing. But these incoming pain and temperature fibers are going to be all over the place. And we're going to get some proprioceptive fibers as well going up into the primary nucleus for proprioception of mastication and things like that. But here, this is the portion that we're talking about now today in the medulla. And we're looking at the spinal portion of the trigeminal nucleus, okay? And it goes all the way down to C2, right? And aligns itself with the substantia gelatinosa. All right, we're gonna look at this more. When I outline, when I use these ovals, I'm focusing your attention. That means this is why this slide is up here. We've moved up one section from the last section, okay? Here is the spinal nucleus and the tract of cranial nerve 5, all right? Let's keep going around, keep ourselves oriented. Here's the lateral spinal thalamic. Here's the spinal cerebellar pathway is up here. We're still doing pyramidal decussation, so that has extent. That's another thing I would like you to take away, is some of the, wait to look at the hypoglossal nucleus, and you're going to see it in the lowest sections and the highest sections of the medulla. And when you look at single axial, you think it's a little tiny round ball. It's not. It's a long round column, okay? A long oblong. And a lot of the nuclei are like that. And we're going to cover that in detail. Here's the MLF being pushed out by the pyramidal tract and the decussations. Here's a rubrospinal pathway coming down here. Still got central gray. Look how big nucleus cuneatus has gotten so quickly. Vesiculus, of course, is shrinking. Why is it shrinking? Well, because the fibers are terminating in the nucleus cuneatus. Look how big the fasciculus gracilis, almost gone. Okay, so very good. Let's keep going. Same level, but now we're looking at a Nissl stain instead of a Weigert stain, so you can really see what the cell bodies of the nuclei look like. Here's nucleus gracilis, big neurons. Nucleus cuneatus. Here's the nucleus of the spinal tract of 5. Here's those anterior horn cells in the supraspinal nucleus. Here's a reticular formation. We're going to come back and talk about the reticular formation in some detail today. And here are some of the cells of the central brag, quite tightly pointed together. All right, let's take on a new topic, right? We move quickly, picking up a new topic, the inferior olivary complex, whole new subject in the medulla. So the inferior olivary complex has a principal nucleus and two accessory nuclei, and we need to understand them all. The principal nucleus projects across the midline, runs right through the other olive. It runs right through the contralateral olive and then ascends to the contralateral cerebellum. Contralateral cerebellum. That's a little peculiar. Most of the stuff that we think of are usually ipsilateral, but not everything. So we're for instance, the left cerebral, corticospinal, the left corticospinal has to interact with the right cerebellum. So that's going to have to do that. The upcoming information from the spinal cord is mostly ipsilateral, but we've got a lot of descending information that we'll have to get to the contralateral cerebellum. This is one of the ways that this happens, all right? And the inferior olive gives rise to the climbing fibers. And the climbing fibers are unique in the cerebellum. They are incredibly powerful excitatory stimuli, one fiber per Purkinje cell. During development, there are many, but they are all going to disappear. And in the adult, there is one olivary fiber, one climbing fiber per Purkinje cell. This is an extremely powerful input. And now it will be modified when we get to cerebellum. We will be looking at what are called mossy fiber inputs, which is all the information that the cerebellum needs. But the climbing fibers are not so much information as they are pay attention, get busy, okay, participate in this event. The inputs then to the inferior olive, look, cerebral cortex, pretectum, red nucleus, periaqueductal gray, vestibular nucleus, the spinal trigeminal nucleus, and the contralateral cerebellum are the things that go into the olive so that it can then cross the midline and put that information into the contralateral cerebellum. That would be simple and clean if it were only that, but it gets worse because you have what Alf Brodau called the indirect spinocerebellar pathway. This is also romantically referred to as the double cross. This is the cerebellar double cross, because the ventral spinocerebellar projections from Clark's nucleus, okay, they are crossed in the spinal cord, like the spinothalamic tract. The ventral spinocerebellar projections from Clark nucleus cross within a segment of where they come in, and then they ascend to the accessory olivary nuclei, who then send their information to the contralateral cerebellum. But they've had an opportunity to interact with the principal olivary nucleus and all of its input, okay? And the same thing is going to happen with the accessory cuneate. That's going to cross to the contralateral inferior olivary nucleus and then go right back to the cerebellum. That's why it's referred to the contralateral cerebellum. That's why this was referred to as the cerebellar double cross. Okay, but I like Brodau's term. It's an indirect spinocerebellar pathway. And the major pathways are direct, but this is an important one. Now, here we are, and we put our eyes back on and start, okay. So we're clearly at a higher level, and we're characterized by the pyramids. And you can see the principal nucleus, and you can see accessory nuclei related to the olives. And so let's start here and just work our way around. Here's a tiny little arcuate nucleus. Here's the corticospinal tract, all right? That's where it looks like we're mostly above the decussation now. Here we are at the medial lemniscus, and we are at the decussation of the medial lemniscus. That's what's going on here. This is the decussation right here. Okay, let's go around. Inferior olivary, spinal thalamic tract, root fibers of 12 coming out, hard to see on the Weigert stain. Nucleus ambiguous, so we're going to talk about it. What I just want you to see where the nucleus ambiguous is. We're just looking and looking and looking. Hypoglossal nucleus, right in the midline, because it's a general somatic efferent. So it belongs in the midline, along with cranial nerves 3, 4, and 6. Those are the only true somatic motor in the brainstem. Here, further out laterally, and we're going to deal with this concept, but further out laterally is the general visceral efferents of the dorsal motor nucleus of cranial nerve 10. We're going to deal with it. Here's accessory cuneate nucleus. Here's the dorsal sensory nucleus of cranial nerve 10, still further lateral and posterior. Because why? Because, you know what, if this was a central canal, then we could draw a line and everything ventral to it would be basal plate and everything dorsal to it would be alar, embryologic basal. Okay, and the wing, the alar, is spreading its wings up here. Here's nucleus cuneatus. It's like, wow, this is all now mixed in with fasciculus. Here's nucleus gracilis, no more fasciculus. Here's the area postrema. We're going to look at that shortly. Here is a very pretty picture of the nucleus and the tract of the solitary nucleus and its tract. Very nice. Solitary nucleus and its tract. Good slide, okay? Here's the descending nucleus of 5, getting tired of that. Here is the MLF where it belongs, finally, okay? All right, and so this is going to be heading up toward cranial nerve 3 and 4. Here's the lateral reticular nucleus. We're going to divide the reticular nucleus into its 3 parts. Here's the internal arcuate fibers. What are they? Well, they're the crossing of the fasciculi of the medial lemniscus, really, is what they are. Okay? You love it? We love it. Speaking of the area postrema, yeah, not a neurologist's favorite structure because we feel so bad for our patients when they have lesions in the area postrema because they're sick as a dog throwing up, which is what the area postrema is for. It's actually there to cause you to throw up. Why would you want to throw up? Well, if you eat the wrong thing, you want to throw up quick, okay? That's what you want to do. By the way, it's attached to a wonderful memory device. No matter what the food was that you ate before you threw up, you will not want to eat that food again. Whether it had anything to do with why you're sick or not, my son never ate a hot dog again, okay? Just never ate it again, and that was all there is to it. It's attached to the chemoreceptor trigger zone, which is really part of the area postrema. This is outside the blood-brain barrier. It's part of what's known as the circumventricular organs. It has direct access to CSF, and it detects something that it doesn't like, and it doesn't have a blood-brain barrier. So if it detects something that it doesn't like, okay, you're going to throw up, and that's all there is to it. I remember once giving a guy with an opiate overdose a cup of what do you call that? Of the oh my gosh. The emetic, the very powerful emetic. Well, what is that called? We used to Ipecac, Ipecac. So, and I give him the cup of Ipecac, and he holds it, and he says, "I know what this is." Doesn't take it, doesn't taste it, smells it, and starts vomiting uncontrollably. Yeah, it was amazing, just from smelling it. All right, and the memory. It receives input from the solitary nucleus on the spinal cord, and it projects to the solitary nucleus, but also to the locus ceruleus, and also to the dorsal motor nucleus of the vagus. Why would it project to the dorsal motor nucleus of vagus, hypoglossal, mesencephalic 5? Okay, why would it do that? Yeah, exactly. because that's how
is everything coming in the wrong direction?
Yeah, we got to make this happen. So in order to make it happen, it has to have access to all those motor nuclei, and it does. All right, and we're going to look at all of those nuclei. And here's a picture of area postrema, and here it's not the initial colliculus or what do you call these? prominences. At any rate, it's the second one. This is hypoglossal, and this is the aericostream. All right. Here, by the way, here's cranial. This and this little blow up. We can look at this. This is all that general somatic sensory information way out laterally. Okay, and in here we're in the. This is the solitary tract and the nucleus of the solitary tract. So you can turn it around the other way. Dorsal motor nucleus, vagus, cranial nerve 12. Nice, nice slide. Lots and lots of nice slides to help us. Okay, I want to get on to the medullary reticular formation. And of course, the word reticular is net, right? I mean, that's what rete, so it's a net. And it's a diverse aggregation and a complicated network. And I really think that this goes well if you will think of the sea anemone, all right? And I'd like you to think of the brain of the sea anemone. So in fact, the sea anemone has no brain at all, okay? But it has this network of neurons which are interconnected, because the anemone is capable of a lot of things. It has arms that move the water in, so it has stinging tentacles. It's capable of a lot of things. And it has this network of neurons which coordinate all of the activity. And I think it is really useful in the human to think of the reticular formation as just that, as this network of neurons which are going to execute many functions in a coordinated way, which are accessed by our descending influences. And they're going to take control of the reticular formation. This is extremely important when you get to eye movements, okay? And when Terry Hammond Patterson was here last year or the year before, we were discussing the pathogenesis of ALS. And a very important question is, why are the ocular motor muscles, why are they not ever involved in ALS? Never. What is it? They're true somatic motor. Why in the world would they not be? And the answer is that there are no cerebral descending nuclei, corticobulbar, corticospinal tract, that ever contact cranial nerve 3, 4, or 6. The 3, 4, and 6 are contacted indirectly by the reticular formation. And the descent, when we want to look in a direction, we access the reticular formation. We never touch the cranial nerve nuclei for vision. That's an extremely important principle, okay? And we do that over and over again. You don't want to know, right, that you have to think to keep your eyes pointing in the same direction, or what muscles you need to activate to sniff or to sneeze, okay? You don't want to think about it, right? That would be a waste, okay? So you don't do that. You simply activate the reticular formation. Who is capable of doing it, has been doing this since the sea anemone, okay? That's what I would say. That is the way to think about the reticular formation. All reticular neurons project. This is an old system. See, if you think of it that way, it starts to make sense. These neurons project rostrally and caudally. Okay, they don't care. It's not like the modern systems that we're used to. They have long radiating dendrites that go all over the place. This is a system which is designed to integrate all of these primitive actions. You have in the, of course, like everything with development, with ontogeny, comes the added complexity. And so in the human, the reticular formation, mammals, the reticular formation takes on divisions and things like that. It gets more complicated. It's not the simple picture that I'm giving you would not be accurate in detail. It's just the concept. So you have in the human, the nucleus reticulus gigantocellularis, which are big cells. They receive input from the cortico-reticular pathway. That fits. They project to the intralaminar nuclei of the thalamus. We're going to do thalamus in total detail, right? Late, all right? And we're going to look at the intralaminar nuclei, and they're very important. And their descending, by the way, their descending influence paralyzes us. They inhibit all of the spinal motor neurons simultaneously as a network, And if that isn't designed for REM sleep, I don't know what is. Okay, so, right, because during REM sleep, we're all paralyzed. So how do you do that? In order, any function that occurs, okay, has to have a piece of the nervous system that does it, all right? So there has to be a system for paralyzing us during REM sleep. This is it, all right? The lateral reticular nucleus projects to the cerebellum and has input from spinal reticular pathways with large receptive fields. And then you have small cell groups that get vestibular and auditory information that are going to project to the cerebellum. Okay, we're just looking at the reticular formation. We've seen this slide. Yeah, we've seen this slide already. Here's a reticular formation. It's loose. You can't, it's very hard to identify. We're going to see some slides that make believe that you can identify it all. And here you are, and let's stay focused on the reticular formation. By the way, down here, I put the nervous system of the sea anemone, just to make a point. I just put that in last night. And here you go. Here are the raphe nuclei of the reticular formation. Here are the, geez, these are large cells. Oh, this must be gigantocellularis of the reticular formation. And here's the lateral reticular formation. Those are very small cells out there, obviously. And then I like this slide. And you can see here the raphe. Okay. And then the perivascular, here's nucleus reticulus. Yeah, portus cordalis, that's not what I'm looking for. Here's raphe nuclei I'm looking for. Here's the small cell group of the reticular formation. And then here's gigantocellularis. That's what I was looking for here. Obviously, I'm going to ignore that. Now, shift. We're just covering lots of different topics in the medulla, very big and complex structure. The ascending and descending tracts that are running through the medulla. You have, in addition to the ones that we've discussed, you have a spinothalamic, all right, the spinothalamic tract. That's going to decrease in size. Why would the spinothalamic tract decrease in size as it runs through the medulla? Because as neurons jump off and synapse onto reticular formation neurons, so they're not going to continue on their way to the thalamus. So although we call it a spinothalamic tract, inside the spinothalamic tract is a spinoreticcular pathway as well, embedded within it as they leave the spinothalamic tract macroscopically. It's going to get smaller. All right. You have the dorsal spinocerebellar tract, which will go through the inferior peduncle. You have the ventral spinocerebellar, which does one of the weirdest things imaginable. It's going to go all the way dorsal, and then it's going to actually enter the cerebellum via the superior cerebellar peduncle. It's like, I don't understand that. We will get to that, not to understand it, but just to deal with it in detail when we talk about the cerebellum. The descending tracts that run through, we talked about the MLF as a descending tract, and that was a surprise. We have a rubrospinal, which are jumping off. You got lots of rubroreticular neurons, lots and lots of those, okay? And then some of them will continue on their way down into the spine, okay? This lateral reticular nucleus, we said, goes to the contralateral cerebellum. That will allow the rubrospinal tract to come down and then project to the contralateral cerebellum. And we'll have rubrobulbar fibers going all the way down to the inferior olive. We're not going to look at this slide right now, but I put it in here because it's a great review. This is a wonderful slide. Not only is it a good section and colored to help you, but not only it's labeled and shows you the projections in and out. So this slide is a wonderful review slide, okay? But we're not dealing with it now. Okay, moving up now. Let's just go around again. What are we looking at? All right, let's start down here, I guess. Here are the pyramids. Here's the medial accessory olivary nucleus. We saw that before. Here's the arcuate, is so small you can barely see it. Here's the root fibers of 12 emerging between the olive and the pyramid. Here's again the inferior olive, the spinothalamic tract, the olivocerebellar pathways moving up. Okay, some of cranial nerve 5. Here's the accessory cuneate nucleus, right? That replaced Clark's column, all of that. Now we're talking about an inferior vestibular nucleus. It's like, aren't we in the medulla? And isn't that pontine? And the answer is yes and no, because truthfully, the vestibular nucleus sits at the pontomedullary border. It's in and out of both. Here's the dorsal longitudinal fasciculus connecting a lot of vestibular pathways. Here's some choroid plexus, fourth ventricle, more nucleus, okay? Here's the dorsal central nucleus of 10. Here's the solitary tract, all the way around again. Briefly, the inferior cerebellar peduncle is divided up into two parts. There's the restiform body and the juxtarestiform body, and they're very separate. Most of the time, when you think of the inferior cerebellar peduncle, most of the time, you're really thinking about the restiform body, okay? The juxtarestiform body we can dispense with cognitively very quickly, and that is, it's a reciprocal pathway between the vestibular system and the cerebellum system, direct, okay? So you're going from vestibular to cerebellum and back out again, and the cerebellum to vestibular is larger, it's a larger component. This introduces another thing that we're going to return to in a minute, and that is some of the descending influences onto the incoming sensory information in the medulla and spine. And you might think that the things coming in are just, they come in, maybe you organize them a little bit and then you send them north for processing. No, it's not like that, okay? Perception is an active process. If you want to feel for keys in your pocket, you're exploring in your pocket. It's not, you're not just sort of passively accepting the information coming in here. You're feeling for shape, appropriate, you know, this object recognition, right? This is, you're feeling around for shape and that sort of thing. It isn't just a matter of the proprioception. You've got to go find the shape and interpret it. So you have to have descending influences on your incoming information. The incoming information, I don't care whether it's auditory or the auditory is if there's any exception, it's going to be auditory. It comes in clean, relatively speaking. But everything else, vision and all of the somatosensory are coming in already highly processed by focus of attention, okay? So you got to get used to that. And so cerebellum is strongly influencing the vestibular information before it's sent out, or maybe as it's being sent out. It's all of this is ipsilateral in the juxtaversiform. And the restiform body is everything else. Right, you have reticular nuclei, the inferior olive, the cuneus, the accessory cuneus, Clark's column, the trigeminal nucleus, the arcuate nucleus, all of this stuff. And by the way, the arcuate is dealing, we're going to talk about respiratory in a minute. All of that is going through the restiform body of the inferior cerebellar peduncle. So let's just take a look at that. Here we have the juxtarestiform body. There's restiform body. There's juxtarestiform body. Okay, so vestibular. Juxtarestiform, vestibular, vestibular cerebellar, vestibular cerebellar, vestibular cerebellar. Here is the inferior cerebellar peduncle, and this is really the restiform body, okay? And I'm going to skip all the rest of this, and you can do it yourself, but you can see how high we are. But we have a lot of ground yet to cover. Here we come to this concept of this organization of the cranial nerve nuclei. This goes all the way up into the midbrain, but it's easily seen in the medulla. And so the idea is at the midline, you have a general somatic efference. And as you start to move off the midline, move laterally, you're going to hit the general visceral efference, the M10. Okay, then you get to the general visceral afference, so like a book reflected. So somatic, visceral, motor, visceral, somatic, sensory. Okay. And then you superimpose on this special visceral efferents that are going to cranial nerves 9 through 11, and the nucleus ambiguous. And then the special afferents coming in, that is taste, and some of the cardiorespiratory stuff that's going to be coming in. And then you have the totally separate and special somatic afferents that are coming from the auditory and vestibular nuclei. So this is the organization of the cranial nerve nuclei in the brainstem, and particularly in the medulla. And this slide just sort of makes that for you. And here we are, hypoglossal, and here's a dorsal motor nucleus, and here's the dorsal sensory nucleus. If there's a dorsal motor nucleus, there's a dorsal sensory nucleus, right? And then your way out into the specials. all right? And we already talked about nucleus cuneatus and gracilis really are part of this general general somatic afferents, okay? Here's cranial, the vagus nerve, and that's really the way to understand then the multiple components of the vagus nerve is through this organization, and that's the way it works. And you need to study these on yourself. We're in a minute, we're going to be turning our attention to the solitary tract here, okay, solitary tract here, okay, and the nucleus ambiguus here. Also, this is a great slide to give you the sense of the extent. Let's start with the trigeminal complex. It's already going down in the spine, goes all the way up into the midbrain. Huge. Look at it in comparison to the other cranial nerve nuclei. It's enormous. Here's this yellow represents some of the autonomic, the viscerals. Here's the dorsal motor nucleus. Skinny little thing. Here's the hypoglossal nucleus. Remember we said covers most of the distance, axial distance of the medulla. Okay. Here we are dealing with some of the spinal parts. This is the solitary, I believe, or ambiguous. This will be nucleus ambiguous. The labels in there someplace. And the solitary fasciculus is here. All right. Hypoglossal nucleus, not much to say that we haven't already. It's general somatic efferents. It's typical multipolar, like an anterior horn cell. I mean, that's what it looks like. If you looked at the neurons, it look like anterior horn cells. We've talked about its extent. You understand that it innervates the ipsilateral half of the tongue. It emerges between the period. We've talked about all this stuff. It receives, here's an important question now. It receives extensive projection from diverse neurons of the medullary reticular formation and discrete input from the cortical bulbar pathway. Okay? So lots of medial reticular and discrete cortical bulbar. And my question is, why? Why does it have these two inputs?
To avoid suffocate ourselves with our tongue
So
while sleeping.
Yes and no, but let's go toward teleology. What is it doing? What is we're talking about the tongue? What are the functions of the tongue? So here I'm going to give you a quote from pediatric rehab. Okay, a pediatric rehab speech therapist will say to you, the same tongue that pulls the peanut butter off the roof of the mouth also says the word peanut butter. Okay, so and so there are certain things that the tongue is going to do which you don't want to think about. It projects in vomiting, right? It's part of the cough, right? But it also is necessary for our speech. When it's throwing up, it's being run by the reticular formation. When it's pronouncing "I love you," okay, it's the cortical bulbar tract that's running. And you're going to see that with all of the oropharyngeal muscles. That same principle now is going to apply to all of the oropharyngeal muscles because the same ones that gag, okay, you say the word clown, all right? Okay. So here now, nucleus ambiguus. So the term ambiguous refers to the fact that it's in multiple cranial nerves. There's nothing ambiguous about finding it on a select section. There's nothing ambiguous about what it does. It's a simple motor nucleus. It comes like the face and like muscles of mastication. Its innervation is branchial arch, okay? So it's not in the midline, okay? And these branchial arches contribute to 9, 10, and 11, and all of the muscles that go out 9, 10, and 11, with the exception of the spinal accessories, right? All of those that are coming are going to be running through nucleus ambiguus. It's just that simple, okay? Let's not make it more complicated than it needs to be. You want to get into the individual muscles? Fine, go ahead and get into the individual muscles. But the nucleus ambiguus is a simple, general, visceral efferent, okay? That has to do with the oropharynx, period. And that's all there is to it. It's going to get reticular input, it's going to get local sensory input, and it's going to get cortical bulbar input for the same reasons that the tongue does. And that's basically really all that I want to say. And here's, again, here's nucleus ambiguous is here. This is nucleus ambiguous. Okay. And it looks a lot like the dorsal, like the hypoglossal nucleus when you do it on this simple level, different derivation. The solitary nucleus has to be divided in two. There's the rostral division and the caudal division. So that's a peculiar thing. You call it the solitary nucleus and then immediately divide it in two. That's strange. Anyway, the caudal division is viscerotopic. The caudal division is a general visceral afferent. You see how the system really works in the medulla? This really gets you organized. And the caudal division of the solitary nucleus is going to get a lot of visceral input. I mean, I don't know what else to say. Cardiovascular input, including from the carotid, is going in there. Respiratory input's going in there. And digestive information, all going through the caudal division of the solitary nucleus. It's going to project, right, exactly where it should, which is to the dorsal motor nucleus of the vagus and down into the spinal cord to access the respiratory muscles, okay, as well as project upward to the hypothalamus. So this is really integrating. It's an integrative site of cardiovascular information, cardiorespiratory, cardiovascular. We'll look at it in a little bit more detail in a minute. The rostral division is the gustatory nucleus, period, okay? It gets the special visceral afferent to taste as a second order neuron. Nothing primary enters at this level, all right? It's going to get information off the inferior ganglion of 9 and 10, as well as from the geniculate ganglion. And it, of course, is going to project to the thalamus, as well as over to nucleus ambiguous and cranial nerve 12. Great slide, okay? Here's the nucleus ambiguous. Here's the solitary nucleus. Incoming information, cardiorespiratory. Going to go up to next week. We will talk in the pons about the parabrachial nucleus, a major integrative center of cardiorespiratory function. But then the rostral division has nothing to do with that at all. These columns are just aligned with one another, and up it goes into the thalamus. Okay? Dorsal motor nucleus of the vagus, I think we've talked about plenty, and you understand that this is this is your third pigmented structure in the brain, right? Substantia nigra, locus ceruleus, dorsal motor nucleus of the vagus. And it is certainly involved in a big way in Parkinson's disease, and is the reason that the Parkinson patients have all of this dysautonomia. All of it has to do with the degeneration of dorsal motor nucleus of the vagus. Okay? Just showing you some of the inferior and superior ganglion coming in 9 and 10. That's all I want to show you here. Again, spinal root of 11 is a different matter altogether. Here is a fluorescent stain of the dorsal motor nuclei neurons. Okay, sitting very close to the ventricular system. Solitary. Nice, right? Very nice. Solitary tract. Yeah, makes sense. Respiratory center. The solitary nucleus, the caudal division of the solitary nucleus, the nucleus ambiguous, okay, the medullary reticular formation. Put these things all together and you have a respiratory center, both for in and out. They are activated by direct chemical changes, which are transmitted through the vagus nerve. They are associated with the parabrachial nucleus and the pons, right? And again, put together with the dorsal motor nucleus of the vagus, all of this puts together into a cardiorespiratory network, reticular network. There's the red is the cardiovascular center. The purple here is the respiratory center. The solitary nucleus, you're only seeing the rostral division here, but you're really interested in the caudal division down here, that this, and up here in the parabrachial, these are your cardiovascular respiratory centers, okay? Sorry. Cortical bulbar fibers coming down onto the cranial nerve nuclei. These are analogous to the cortical spinal tract, except what's different is that they are usually bilateral. I rarely want to elevate one palate as part of swallowing and not the other. So it wouldn't make much sense to do that. So again, it may bilaterally innervate the muscles directly, or more likely, it's simply going to activate the reticular formation and let the reticular formation, see, Just let the reticular formation do the work. Why should it do this? You could think of it as the way a CPU accesses a screen chip, right? Why should it put each pixel into each chip? Why shouldn't it just tell the video chip what to do? It makes much more sense, right? That's the way this system is organized. And we've talked about this. We're running out of time. Our sensory relay nuclei, you get it. We've talked about them too. But what the point that I want to make here is what I was saying before. That is, you have cortical bulbar descending information coming out of both the pre and post central gyri. So this is cortical bulbar tract, right? It's not going to any motor structure at all. It's going down onto the input sensory nuclei to modulate the incoming information at the level of the second neuron, not the primary neuron, but the second neuron. Okay, let's just say that again. It is known that 25% of the corticospinal tract is sensory, not motor, sensory. And it's not, of course, bringing sensory information. There's no sensory information to bring. It's shaping the sensory information that comes in. I want this. I need more information from the tip of my index finger, and I'm really not caring what's going on my ring finger right now. Okay? So don't send me that information. I don't want it. I'm focused on my pincer at the moment. Shut the rest out. I don't need to hear it. I want to shape that incoming. Sensory perception is not passive. Perception is active. And it has to do with attention. And we're going to get to all of that at the end of the course. We're going to be looking at these mechanisms at the high cortical level. And finally, we have reached the medullopontine junction. So we know that we're close to the end. So what's going on? Well, changing in the guard. We're going to change cranial nerves, right? So we're going to do that. The inferior cerebellar peduncle is going to check out. It's going to go out into the cerebellum and disappear. The olives are going to disappear, all right? The corticospinal tract will become embedded in the cerebellar nuclei, which is going to be interesting. We're going to look at that in a minute. And the reticular formation gets bigger as we move up into the pons and have much more things to do with our reticular formation. And so that's what's sort of going on here. We're still in the medulla. So we've seen this slide before. This is the one that we looked at. But now I just want to focus you on, remember the arcuate nucleus? At one level, we could barely see it. Now it's a capsule around the pyramid. And here it's getting thick. So what's going on with the arcuate nucleus? Here's the rest of the medial meniscus is finishing its decussation. There's the MLF up where it belongs. And then we're going to get into a bunch of stuff that I don't want to talk about today. We are now at the junction. We are getting there, right? So the olive is still there, but definitely getting smaller. The medial longitudinal fasciculus almost looks like it's increasing in size. It starts to gain vestibular and cranial nerve 6 information. Okay, we're getting into cochlear stuff and vestibular stuff and that sort of thing, which we're going to deal with next week. We don't have to deal with it today. And here we are. And this is sort of medulla. And here's the panniculus, if you will, the big fat belly of the basis pontus, sitting over, hanging over the medulla. This guy's used to be the arcuate. And it's huge. It's suddenly huge. And it's a collection. Here, what I like about this is in this panniculus, if you will, of the basis pontus hanging over the medulla is the corticospinal tract. Is still corticospinal tract. Okay, and the cerebellar nuclei are all clumped together. But if we went up to the next slide, which we will do next week, we're not going to have this separation. They're going to be intermingled, giving the basis pontus its characteristic laminar striated appearance in the basis pontus. Okay, so next week, pons. All right, and thank you. And if you don't spend an hour on this, I'd say you're making a mistake. See you next week.